Hi, uh, thank you all for being here, and thank you for the organizers to selecting my abstract for talks. It's my pleasure to tell you what we have been doing. So, I'm a postdoc in Torsten Schwede Lab. So, if you do homology modeling, you may know Swiss model, and so I will tell you what we have been doing, how we have been leveraging AlphaFold predicted structures to increase interpretability of all of the data that now exists. So there was already a bit of an introduction about this in the morning in the beginning of the session, that when you look at protein sequences databases, there's a huge increase on data. And if you look at how they are named and how they are annotated, actually about half of, like 60% of them, are either annotated with data that comes from experiments about these structures or based on homology. 20% are partially annotated, and then there's about 20% of them that just don't have any kind of domains or family annotated over them, and these are the ones that are just called uncharacterized proteins, hypothetical proteins, proteins of a non-function, and so on and so forth. And if we divide this into different species and look at proteomes, actually this distribution is biased, and there's species that we know much more about or can annotate much better, like the human proteome, than others, like something that comes from the environment. And now with all of these big data, like all of the structural information that comes from AlphaFold, I started thinking, okay, how can we help shedding light into these proteins at a large scale? And the way that I like to look at this is by taking a step back and look at all diversity of proteins as a vast universe. So there's the concept of the protein universe, and here proteins that are sampled by nature, or that were sampled by nature, and that we can pinpoint about something what they do, would be bright stars in the space, and then protein families would be galaxies, protein superfamilies, clusters of galaxies, and so on. And you see where I am going to. And then all of those sequences that were not sampled by nature, or that were sampled but we don't know anything about them and we don't know what they are, they would be in these dark regions of the protein sequence space. And when we're trying to get knowledge about individual proteins, you'd select your favorite protein and start characterize it. You're basically selecting a star in one of these galaxies, and then based on homology, you expand it to the entire galaxy and cluster of galaxies. And now, thanks to AlphaFold, we have structural information for not only galaxies that are bright and illuminated, and so we have some idea of some function associated to them, but we also have structural data about proteins that are in these dark regions. And so I wanted to know first, okay, how much of this darkness is covered by the AlphaFold database? How much of it is at high predicted accuracy so that these structures can be leveraged for predictions? And how can we then use that to model this landscape to help finding where are all of those dark proteins in UniProt? So let's go to first the darkness coverage within AlphaFold. So what you see here is a series of histograms. Here, the first one on the right, on the left, on the right. Yeah. Anyway, there. Either on the right or the other right, doesn't matter. So here basically is a distribution of what we call functional brightness, which is a very general term invented by me in this case, which is just how well. So if you take a UniRef50 cluster and look at all the proteins within this cluster and see how well they are annotated at full length with domains and families and predicted coil and predicted disorder regions, you just take the protein that is the best annotated, that would be the brightest in the cluster, and then just expand all of that brightness into the entire cluster. And so here what you see is that if you take all UniRef50 clusters, there's about 20% that there's at least one member in this UniRef50 cluster that is annotated at full length. But more importantly, there's 34% of UniRef50 clusters are composed of proteins where there's no annotations like that, like annotations for domains and predicted disorder and coil in any of the proteins. So these would be the fully dark UNIRF50 clusters. And so if you then select only those UNIRF50 clusters where there's at least one member with a model in AFDB, there's a reduction, of course, in the number of clusters, because there's all viral proteins and proteins in UniPark are not included in the AlphaFold database. And you also see that this bar of darkness decreases. So there's already some UNIRF50 clusters that are composed of proteins that are in this PIN, and they are solely from that. So that information is not, that darkness is not in the AlphaFold database. Then if you take only those UNIRF50 clusters where there's a member with a PLD, with the average PLDDT better than 70%, darkness content decreases again. And if you put that threshold to 90%, it decreases further to 15%. So you go from a proportion of 34% UNIRF50 clusters that are fully dark to 15%. So there's a lot of these dark proteins that are lost in these filtering. So there's dark proteins that are in dark UNIRF50 clusters where AlphaFold cannot really help at the structural level. But most importantly, there's a big fraction where AlphaFold structures can already be leveraged to learn something about them. And these are the ones that we focused on. And this corresponds, so here when you go to UNIRF50 clusters, there's about 50 million UNIRF50 clusters. This reduction reduces the clusters to 6 million UNIRF50 clusters. So it's 10% of UNIRF50, which corresponds to about 50 million UniProt sequences. So then to see how do these, how to try to find how these proteins distribute in sequence space, what we did is to construct a very large sequence similarity network. As far as we know, this is the largest sequence similarity network ever constructed. What you see here, like this, just as it was saying this morning, conceptually, this is really easy. We just did all against all MM6, but it took us 2 years to know how to visualize this and how to better interpret it and make it useful. Here, what you see is a summary of the network that we got is actually a network of connected communities in the very large network. What you see here is about 600,000 nodes. And each node is a community of interconnected, very similar UNIRF50 clusters. And they are mapped, they are colored with functional brightness, so following the same color code as before. So if it's purple, it's functionally dark. So everything in that cluster is not annotated at all. And everything that is white there's at least members that are very well annotated. So you could try to already use some homology to try to do some transfer of annotations. But the important thing is that you see here 2 regions in this landscape. There's what I call the blob in the middle. It corresponds to 50% of the nodes in the network. They are all interconnected. And this is because there's domains that are shared between proteins. There's functional motifs. So yeah, you're going to get a lot of interconnection between different families. But there's also a bunch of different, we call them galaxies because we're looking at the universe, right? So there's a bunch of galaxies around the big blob that, as you can see, it's where most of the purple is, where most of the darkness is. So if we zoom in into the network, we have something like this. Sometimes you have very bright clusters. So here, I don't know, you don't see the mouse. So here where it says bright clusters over there, so there's the zoom. Right, and then there's the bright clusters. It's pointing into something. Actually, it seems like it's empty, but there's just white dots in there. That would be a family or superfamily of where everything can be easily annotated. But then there's those clusters where everything is dark. And so those correspond to the unknown protein families or superfamilies that are composed of these proteins that we don't know anything that they do. But you also see that there's a purple distributed very near brightish points. This means that they are just remote homologs, most likely, of proteins that can be well annotated. So we can already use this information to help the annotation of these proteins. So what we see from this is that actually this is a very rich source of new unknown protein families. And one example is the cluster 159, which I don't have a reason to choose this cluster. I was looking for an example for the poster at the ECCB last year and just came across this cluster, looked at the predicted structure. There was no match to anything in the PDB similar to that structure. And then I ran DeepFry and got to predictions for the structures for the proteins in this cluster for DNA binding and hydrolase activity. And then that was it for the ECCB. That's how much I knew about it. But then after ECCB, I just started digging more into this cluster and constructed a higher resolution sequence similarity network for the members of this cluster, which you see here in the middle, and actually could show that this is not a family, it's actually a superfamily of proteins that are encoded in a conserved, so they have a conserved genetic context. So they are all from prokaryotes. You find them in bacteria and archaea. And the genomic context as is conserved at some point, like there's a level of conservation and then there's a level of non-conservation. So as you see here, basically there's a conserved bicistronic arrangement, and each cluster in the map has its own different neighbor in the genomic context. And when I was discussing with Tanel and Vasily, so Tanel at the University of Tartu in Estonia and Vasily in Lund, I showed them this and they said, well, this looks like a toxin-antitoxin system and we can very easily, hopefully, validate that for you. And that's exactly what happened. This was an incredible experimental collaboration because they could show it in 2 months that this was actually a toxin in a new superfamily of toxin-antitoxin systems. And I will not enter the details, but basically what you see here is the toxicity assays where they demonstrate the toxic activity of the purple, so the members of cluster 159, and the antitoxin effect of the cognate. And here in the bottom, you have metabolic labeling that just demonstrate that the effect is over the incorporation of methionine, so the production of protein. So the hypothesis is that members of component 159 are a superfamily of translation targeting prokaryotic toxins, and we call them now TUM-E for dark in Estonian. But there's much more there. And then at the time that we were analyzing these clusters, there was the new feature on UniProt that they were using ProteinLM to name all of the proteins that are called hypothetical in UniProt. And we thought, well, if these are truly new protein families that don't look like anything that was ever named, maybe this language model will just give a random set of names to the truly dark ones, right? And that's exactly what we saw. So we looked at the distribution of words that were in each of these dark clusters and compared that to the bright clusters. And we saw that there were many clusters, many components, dark galaxies in the landscape where the diversity of words that in the names of all the members in that cluster were much higher than those in the bright ones. And so just put a cutoff of 20%, and together with Alex and Tony from PFAM, now we are incorporating them into PFAM, and they are curating them. And indeed, many of these are true new protein families that now have their own DUF. So this is a huge, big source of protein families. Then we also, as we're dealing with structures, we were also wondering which kind of novelties from the structural level were in there. We didn't do all against all clustering at the structural level because, as was already told, Martin and Pedro were doing that. So we went in another direction and just looked at the distribution of substructures in the dark. So if you break the protein structure into substructures, into local folds, and then compare the distribution of these local folds in our dark galaxies with those in the PDB, we can have a score of whether these proteins are look like things that are in the PDB, so they are inliers, or if they don't, and so they are outliers. And what we saw is that most of the dark components, dark galaxies, are in layers, while when you go into brighter clusters, they are mostly outliers. But there's also some bright clusters that are outliers, and those cases are like repetitive proteins or proteins that need to be in an oligomer to have their proper fold, or even fragments. So these are all bright clusters. They are all members of bright clusters, but they are outliers because this kind of stuff is underrepresented in the PDB. But more importantly, we could find some novel folds, and one example is these what we name now the beta flower, and together with Tony and Alex, and now trying to figure out what these proteins may do, what are their diversity, how widespread they are, and etc. We have this landscape, or this this network available as a web resource, which we call the Protein Universe Atlas. You can go to uniprot3d.org and navigate through the landscape. It is interactive. You can search by, you can see here, you can color it based on different metadata. You can also select for the clusters based on the metadata that you want. So if you want functional darkness, or if you want the semantic diversity, you can search by uniprot AC, you can search by component AC, and you can also, so you see if you just search something, you have a table, you can navigate through, you can download stuff, you can click in the nodes and get information about what is there. You can see the structures, the predicted models for the member in that cluster. And now you can, I just want this to move forward, because you can also query by other things like protein sequence and protein structure. So you can now also search with protein structure, and it will tell you, so this in on the side, there's other hits that are the clusters that are hit with your search, and then you can see where it falls in the network, and then from there you can decide whatever you want with those results. 40 and then we plot the same graph. If you're interested from the structural perspective, I highly recommend you to check Martins and Pedro's web resource on the clusters from the structural perspective. And with this, I'm finished. I hope I have convinced you that this is a nice approach to, at large scale, find new protein families and navigate through the huge diversity of sequence and structural data available now. Of course, in our plans is to now expand this to much, much more data, but we're working on ways of dealing with the network now. But what we think is very obvious from the kind of works that are emerging now and with our work is that as a community, we are closer and closer to be able to explore all the unknown territory of the protein universe. So I want to thank you all, but I also want to give a very big thanks to the Schwede group, and especially to Torsten, to host me as a postdoc with this crazy idea of making a model of the protein universe. And there's other people from the group around. So there's me, there's Lorenzo, Gerardo, and Arthur. So if you want to learn a little bit more of what's going on in the lab, also talk to them. And I need to thank the collaborators in Estonia and Sweden for the experimental validations, to Alex and Tony for jumping in and helping with the analysis of all of these clusters, to Mehmet, which together with Janani developed the shape descriptors for the proteins, to Scicor for computational support, and all of the other people that some of them are on the audience with whom we have been discussing about this project and have given very nice insights on its development. We have a preprint and also a poster today, so if you want to discuss more, come to me. Thank you.
Thank you.
Hi, I'm interested in the shape of your universe. So to my mind, I mean, on one hand, it looks like penicillin mold growing on a pancake, right? But there's two distinct morphological kind of things that come out of it. So there's the penicillin-y bit in the middle, and then there's the kind of peppery bit around the outside of dots. What I assume this is some artifact of the way you've plotted it or connectivity. What's the reason for that morphological distinction?
Yeah, so this is just an artifact of the layout. So we use a force-directed algorithm to lay out it. And of course, everything that is interconnected would be pulled to the middle, and what it's not, it will be repelled to the sides. So that's why we have this blob in the middle, and they lack connection to the remaining things. So they connect between them. There's like, they are some dots that connect to themselves, but then they don't connect to anything else in the side. So actually their relative position to the rest doesn't mean anything. It just means within the things that to which they have edges to.
Great talk. Thank you. It's a heroic effort, really. But I have a comment. I've worked with this guy, Morgan Price, who's made his career on re-annotating microbial proteins with high throughput genetics and then kind of follow-up experiments. And his view is that annotations are lies, really. And so I'm actually curious if you think there's some kind of application here to check the quality or consistency of the annotations we already have, right? So the labels you're using to propagate and train.
That's a very good point. I actually didn't look at the diversity of annotations within the NRF50 clusters, but that's a very good point. I'll look into it. Yeah.